I'm very happy to be here today and to present the work we've been doing regarding the use of antagonistic pleiotropy and collateral sensitivity mechanisms to target drug resistance in leukemia. This work has been performed at St. Louis Research Institute that is located here in Paris. So drug resistance is a major cause of treatment failure in patients with cancer. We know that over the last 4 decades, many targeted therapies have been developed, and so far most of them will mostly delay disease progression until drug resistance emerges. Here's a typified patient diagnosed with acute myeloid leukemia, or AML. This patient will be treated with 1 round of standard induction chemotherapy to achieve an overall remission of the disease. Unfortunately, in about 30 to 50% of the time, we observe also a phase of relapse, and at this stage, no salvage therapies are available, and most of the patients will eventually succumb to their disease. Since the 1980s, Peter Nowell formalized the idea that cancer evolution can follow an evolutionary process that evolves according to both diversification and expansion of distinct subclones by virtue of natural selection. Since then, many single-cell DNA sequencing studies, whole genome sequencing studies have been developed, and they have been able to track individual genetically distinct subclones to formalize the idea that indeed the tumor evolution is heavily influenced by the selective pressure that is mostly imposed by therapy. Here, for instance, we can observe on this plot where we can follow the different genetic subclones that emerge in a patient with acute myeloid leukemia after chemotherapy treatment or bone marrow transplantation. And we can see that the genetic landscape of this patient is completely different between the time of diagnosis, after chemotherapy, and after bone marrow allotransplantation. So this suggests that indeed the therapy is a very important feature to dictate the tumor evolution. Since then, other studies have also established that drug resistance can also occur by non-genetic selection, mostly through the rewiring of the epigenetic landscape of the cells. So the long-standing view that the cancer evolution can follow the same pattern as natural evolution led us to wonder whether the same mechanisms of population extension as those seen in the natural world could be engineered to drive the demise of the tumor. One such a mechanism of local extension is called evolutionary traps. These traps have been involved in local demise of numerous organisms, from birds to turtles to butterflies. These traps occur because specific traits of a given organism that are well adapted to a given environment can become suddenly maladapted following changes that could drastically appear to that environment. Let's take, for instance, the phenotypic distribution of 2 kinds of butterflies, either black or white. If a strong selective pressure is applied against the white butterfly population, this will give rise to a population that is dominated by the black butterfly population. In that case, this population is well adapted to this new environment. And if we change suddenly the condition of adaptation of this new environment to favor the white butterfly population, which is now already extinguished, this will result in a global extinction of the whole butterfly population in a process that we call an evolutionary mismatch or evolutionary trap. This principle could be readily applicable in the context of drug treatment if, for instance, we assume that a population can become resistant to the treatment with drug number 1. This could set up an evolutionary trap that could be triggered by the use of a carefully selected secondary drug. We could imagine then that we could treat patients with a sequential use of 2 different drug combinations to maximize the effect of these 2 drugs on the remission and minimize the relapse of the disease. This evolutionary traps idea arises from the fact that no single adaptation can be advantageous in every environment. There is a genetic trade-off that is then inherent to these evolutionary processes. The geneticists have formalized this idea through the concept of antagonistic pleiotropy, or AP. These define genes that could exert 2 or more traits that could exert multiple threats with opposed effect on cellular or organismal fitness. This means that the net fitness of an AP gene is directly dependent on the type of pressure that is applied on the cells or on the given organism. Using functional genomics, we can actually study the function of AP gene. We can assume, for instance, we can knock out the expression of gene A and study the effect of this knockout in the context of 2 different drug treatments. If the knockout of gene A is able to increase the cellular fitness in the context of drug 1, but decrease the cellular fitness in the context of drug 2, then this gene is defined as an AP gene. There are precedents in the study of these genes, mostly in yeast, where people have tried to identify AP genes, and they've been able to identify one gene in particular, for instance here, PDR17, as a gene that will, once KO, once knockout, will favor the growth of the yeast in the YPG media, or decrease the fitness of the yeast in the ATH media. Using functional genomics, we decided to try to mine for AP gene in the context of cancer, and more specifically in the context of AML. So we decided to leverage a CRISPR-Cas9-based library that we have infected in one AML cell line called OCI-AML2. And this library was targeting over 2,400 genes that were already described to have a role in the context of drug resistance. These cells were then grown for 2 weeks in the presence of 9 different drug combinations, drugs that were already used in AML or that are currently under clinical investigation in this disease. After this 2 weeks propagation, we then compare the global representation of our CRISPR guide between the DMSO and each of the 9 different molecules. We were then able to identify several classes of genes, genes that were defined as resistors, meaning that their knockout will promote resistance to any of these 9 different molecules, genes that were defined as inert, meaning that their knockout will not affect the cell response in any way, and finally, genes that were defined as sensitizer, meaning that their knockout will increase the sensitivity of the cells to any of the 9 different molecules. Following the minimal definition of an AP gene as a gene that could confer sensitivity to drug 1 but resistance to drug 2, we were able to identify around 1,100 genes that follow the definition of AP gene across the 9 different drug treatments. We could then build this complex circoplot where we could analyze the response of each gene across the 9 different drug combinations, and we could rank our 2,400 genes from the top resistor to the top sensitizer. And then we could also connect the function of our gene across 2 different drug combinations. We could establish here on this breakdown of the 36 different combinations of the different molecules, genes that were defined as common sensitizer across, for instance, 2 combinations of molecules like the cytabine or the cytidine, genes that were defined as common resistors across 2 combinations, or genes that were defined as AP gene. Going back now to this breakdown of the 36 different molecules, we found that one combination of molecule in particular, a combination made of ABT199, also called venetoclax, which is a BCL2 inhibitor, and JQ1, which is a BET bromodomain inhibitor, confer the highest proportion of shared AP interaction in comparison with the 36 other combination of molecules. We found that indeed over 75% of genes, when we compare these 2 molecules, shared could be classified as AP gene. Going back now to this cluster of 2 different molecules, we identify a group of 5 AP genes that cluster together. First, the 2 members of the PRC2 complex, EZH2 and EED. Then NSD2 and NSD3, which are 2 important regulators of the H3K36 methylation mark. And then the transcription factor MYC, which code in concert with NSD2 and 3, but in opposition of the 2 members of the PRC2 complex. So this led us to make the hypothesis that NSD2 and 3 could be positive regulator of MYC, while EZH2 and EED could negatively control MYC expression. To test this hypothesis, we designed 2 different CRISPR guides targeting NSD2 and 3, and defined indeed that the suppression of this target will decrease MYC expression, while in contrast, the suppression of the 2 members of the PRC2 complex increased MYC expression. So this suggests that this gene could behave as AP gene because of their regulation of the expression of MYC. So this begs the question of why. Why a non-oncogene such as MYC could behave as an AP gene? The response to this question could be found in the lesser-known role of MYC as an apoptotic driver. It has been shown in several recent studies that cancer cells can tolerate high expression level of MYC because they could also express high corresponding level of anti-apoptotic proteins such as BCL2 that will help the cells to shunt the pro-apoptotic effect of high expression of MYC to favor its pro-proliferative function. In the context of JQ1 treatment, this model holds true. Overexpression of MYC is able to drive resistance to JQ1 treatment, while its shRNA-mediated suppression increases sensitivity. When cells are now treated with ABT-199, the pro-apoptotic effect of MYC predominates. In that case, overexpression of MYC hypersensitizes the cells to ABT-199, while its suppression increases resistance of the cells. So this suggests that the inhibition of the BCL2 protein using ABT-199 is able to turn MYC from a pro-proliferative driver to an apoptotic liability. Given our previous observation that MYC upregulation could drive resistance to JQ1, we asked whether if cells got naturally evolved to become the resistance to JQ1, they could also express high MYC level. So we decided to derive 5 AML cell lines to become resistant to JQ1, and we found that this was associated with an upregulation of MYC in the context of JQ1 resistance in comparison to the parental counterpart. Interestingly, we also found that across the 5 different models tested, we also found a decrease in the expression of EZH2 that was consistent with our previous observation that the suppression of the PRC2 complex could repress MYC expression. To confirm this observation, we introduced EZH2 in JQ1 resistant cells and indeed confirmed that this was sufficient to repress MYC expression. So in conclusion of this set of data, we found that MYC upregulation is a pervasive feature of JQ1 resistance, and this could potentially set up a trap that could be triggered by the ABT-199 treatment. To test this, we indeed treated our 5 AML cell lines resistance to JQ1 with ABT-199 and found that they were highly sensitive to the treatment in comparison to the parental counterpart. To make sure that this effect was truly MYC dependent, we then suppressed MYC expression using 2 different shRNAs in our JQ1 resistant cells and found that this completely abolished the hypersensitivity observed to ABT-199 treatment. So this suggests that MYC upregulation is indeed an evolutionary trap that could be sprung by the ABT-199 treatment. We finally wanted to apply this idea of MYC-induced antagonistic pleiotropy to try to design a treatment regimen that could be tested in the PDX model of AML. We suspected that the pretreatment of those mice with JQ1 followed by ABT-199 could be a treatment regimen that could allow a more durable disease remission. So we designed a treatment regimen in which we treated the mice with ABT-199 continuously, or JQ1, or switch from one drug to the other. First, we confirmed that cells that got pre-treated with JQ1 and that relapsed after JQ1 treatment are indeed more sensitive in vitro to ABT-199 treatment, suggesting that this evolutionary trap is intact in vivo. Given that the AP interaction never worked in reverse, we then suspected that the pretreatment of the mice with JQ1 followed by ABT could be the most efficacious to treat our mice. And this is indeed the case. When we treated first the mice with JQ1 followed by ABT-199, this led to the most drastic downregulation of the overall disease burden in the bone marrow. So in conclusion of this first part, we found that we could build an empirical and conceptual framework using CRISPR-Cas9 screen to build a rich landscape of therapy-induced AP in AML. We found here a particular example of a mixed-centric transcriptional axis that could be responsible for resistance to JQ1 and hypersensitivity to ABT199. And finally, this idea led us to design a new combination regimen based on sequential use of 2 different treatments to maximize the effect of the therapy in mice. Besides the antagonistic pleiotropy phenomena we just discussed, there are also what we call the collateral sensitivity mechanisms that are also very important in the context of drug resistance. These have been formalized in the study of bacteria, where they've been able to study the resistance of bacteria to specific antibiotics, and people have been able to see that the evolving resistance of bacteria to drug 1 could lead to hypersensitivity to drug B, and vice versa. So we hypothesized that using and combining functional screening approaches with some sort of profiling study, we could also nominate collateral sensitivity mechanisms in the context of cancer. So to do so, we decided to focus our attention on a specific compound called selinexor. Selinexor is an inhibitor of the enzyme XPO1, which is responsible for the shuttling of proteins from the nucleus to the cytoplasm. And according to this effect, selinexor is currently under clinical investigation in AML. We focused our attention on the selinexor because owing to this effect on XPO1 and on the plethora of different proteins that could be affected by the inhibition of XPO1, we suspected that selinexor could elicit in cells both pro and anti-fitness effect for their growth. So this will be the most likely to identify mechanism of collateral sensitivity in AML. So we focus our attention on our favorite AML cell line, OCI AML2, that got transduced using the same methodology as described before, using the 2400 gene library. These cells were then grown in condition of DMSO and selinexor for 2 weeks, and then we compared the global representation of the sgRNA between the DMSO and the selinexor condition. We compared this analysis with an RPPA profiling of these cells that allowed us to establish the set of genes the set of pathways that could be activated or repressed in response to increasing time points of selinexor. So first, we established, using our CRISPR screen, several sets of genes that were sensitizers in the context of selinexor treatment, mostly genes that were related to the AKT signaling or mTOR pathway signaling. And the RPPA analysis was intriguing. Why? Because we established that several phosphorylation sites on AKT were more activated in the context of selinexor treatment, which was a bit counterintuitive because selinexor has killing activity on AML cells, while here it was activating pro-survival pathway. So this will maybe elicit AKT as an important factor to nominate collateral sensitivity mechanisms in AML cells. So first, we confirm across multiple AML cell lines that the selinexor treatment over different time courses will indeed increase AKT activation, as seen here on the 2 phosphorylation sites, threonine 308 and serine 473. And this effect was truly XPO1 dependent because the knockout of XPO1 could phenocopy the effect of selinexor in terms of AKT activation. We then wanted to identify the upstream activator of AKT signaling upon selinexor treatment, and to do so, we had to leverage 2 different techniques that got combined to get a positive hit. The first technique is an RNA-seq study to try to identify the selinexor target genes. The second approach was a flow-based CRISPR screen to nominate AKT activators in the context of selinexor treatment. We took OCI-AML2 cells that we transduced with a library targeting the full genome, and then we treated those cells with selinexor for 48 hours prior to sort by cytometry, the bottom part of the population that are the cells that are possibly activated by selinexor on the AKT signaling, and the top portion of our population that were constituted of cells that, on which the selinexor inhibit AKT signaling. and we're able to identify here in red several hits that constitute the upstream potential target that could activate AKT upon selinexor treatment. We found here 2 positive controls, obviously, of our screen, which are AKT1 and AKT2, because obviously they are constituting the AKT signaling. Oops, sorry. Then by combining our CRISPR screen approach and the RNA-seq study we just described, we're able to identify one gene in particular called P2RY2. It's a purinergic receptor that's scored at the intersection between these 2 approaches. First, this gene was upregulated in the context of selinexor treatment, and he also scored as an AKT activator, also, again, in the context of selinexor treatment. We validated that the purinergic receptor was indeed more expressed in the context of selinexor treatment, here by RT-qPCR, and we confirm also using 2 different shRNA targeting P2RY2 that the suppression upon selinexor treatment will decrease AKT activation. So this suggests that selinexor is able to promote the expression of P2RY2, and that P2RY2 in turn activate AKT signaling. We then wanted to investigate whether AKT inhibition could enhance the therapeutical efficacy of selinexor in vitro and then in vivo. So we started first with the in vitro experiment across multiple AML cell lines. We treated them with a combination of selinexor and multiple AKT inhibitors. And we found that in every instance tested, inhibition of AKT will enhance the killing activity of selinexor. We validated those results in primary patient samples. We found that 99% of the patients tested were all responsive to the combination of selinexor and AKT inhibition. Out of this 99%, 50%, in 50% of these patients, we found this combination being additive, and in the other half of the patients, this combination was synergistic. Given these striking results, we decided to move forward into preclinical testing of the selinexor activity in combination with AKT inhibition in vivo. So we use a syngenic mouse model of AML driven by a fusion oncogene in AML called MLLIF9. Those cells were injected into irradiated recipient mice, and those mice were treated with either selinexor or ipatasertib, an AKT inhibitor, or the combination of these 2 compounds. And we found that the combination of these 2 compounds were able to significantly prolong the overall survival of the mice in comparison to every compound taken alone. This was also associated with a drastic decrease in the disease burden of the bone marrow, of the MLLIF9 blast in the bone marrow of the mice. We then decided to test the efficacy of this combination in a patient-derived xenograft model, the same way we used MLLIF9-driven PDX cells that were injected into irradiated recipient mice, treated again with the combination of selinexor and ipatasertib. And we found that here again we're able to prolong the overall survival of the mice in comparison with every drug taken alone, and this also was associated with a strong reduction of the disease burden in the bone marrow. The last experiment we wanted then to achieve was to compare the effect of the combination selinexor and ipatasertib with the standard chemotherapy regimen that is currently used in human and that we translated into a mouse system. So we use our syngenic mouse model, injected again with MLLIF9 cells. It got treated with a combination of cytarabine and doxorubicin, which constitute the standard chemotherapy backbone in humans. And we compared this with mice treated with selinexor and ipatasertib. So here we can see the results 1 day after the end of the treatment. We see, as expected, that the chemotherapy and the combination of selinexor ipatasertib can achieve a very good remission in mice. But we are very happy to observe that at the time of relapse after chemotherapy treatment on the mice, the combination of selinexor and ipatasertib could still exhibit a pretty potent effect. So all these results taken together, we've been able to combine RPP analysis and CRISPR screening techniques to identify collateral sensitivity mechanisms in the context of selinexor treatment in AML. We have been able to identify a P2RY2 centered axis, P2RY2 being upregulated by selinexor to activate AKT signaling. We found that the combination of selinexor and AKT inhibitors can act synergistically in vitro and in vivo in multiple immunodeficient mouse models. And finally, we found that this combination therapy could, at least in our model, outperform the effect of standard of care chemotherapy. I would like to thank you for your attention. I would like, obviously, to thank all the members of the labs who participate actively into the study, mostly people highlighted here in pink. I would like to thank also our long-standing friend and collaborator in the US with whom we have performed the study, our local collaborator, obviously, at St. Louis Hospital, and elsewhere in France and in the US. Thank you so much for your attention, and I'm very happy to take any question. Thank you.
Thank you.